Welcome back to microbiology. Today we're going to start Unit 10, and Unit 10 is going to cover immunity. So what exactly is immunity? Immunity is going to be defined as a resistance to or the ability to resist infection and disease. It's going to be maintained by an immune system. And it's also going to be dependent upon a variety of structures and mechanisms involving many different types of cells, different types of tissues, and even different types of organs. So going in with the body's defenses, we're going to talk about two types of immunity. We're going to have innate immunity and we're going to have adaptive immunity. With innate immunity, that's going to be defined as the resistance to or the ability to resist infection and disease. It's going to involve body structures, it's going to involve cells, and chemicals that are going to be present at birth. It's going to be considered what we would call like a non-specific immunity. That means it's going to provide defenses against multiple different types of pathogens. It's also going to include the first and second lines of defense, which we'll discuss in a little bit. Adaptive immunity is going to be considered the third line of defense. Now with adaptive immunity, this is going to be one that your body has to learn the organism and has to get used to the organism in order to adapt itself to be able to fight it off. So this is where your antibodies are going to come in. So with innate immunity, these are going to be things that your body's already able to do. For example, a fever. It's going to be able to send out different types of chemicals. It's going to have cells that are going to be like macrophages that are going to be able to go out and ingest these types of things and destroy them. So that's going to be your two different types of immunity. Sorry about that. I'd switched the screen and didn't mean to. All right, so with this, looking at the different types that we're going to have with the innate immunity, you can see these here. We've got mast cells. Mast cells we're going to talk about. They're able to release histamine. That's going to cause inflammation and cause blood cells to rush to an area. You've got dendritic cells and macrophages. You're going to have all these different types of cells that are going to be in your body when you're born that are able to go out and they're able to tackle these different pathogens that get into the body. Now with adaptive immunity, these are going to be things such as antibodies, and those are going to come from your B cells, as well as T cells, which are then going to divide and make CD4 T cells and CD8 T cells. Now going in with our first line of defense, we have the skin. So the role of the skin is going to be twofold, basically. It's going to have a mechanical role and it's going to have a chemical role. Now with the mechanical role, that's going to be the different layers of the skin. So basically it is a protective layer. It's made of two different layers. The top layer is going to be the epidermis. That's the outermost layer, and it's composed of tightly keratinized packed cells. It means it has lots of keratin in it. It's going to make them waterproof, and as you move towards the upper layers of these cells, they are going to be dead at that point. They're going to act as a barrier to most bacteria, fungi, and viruses. You're going to shed the dead cells and remove these microorganisms that are attached to the epidermis throughout the day. So you're constantly shedding hundreds of thousands of cells throughout the day. They're also going to be composed of dendritic cells. These dendritic cells are going to be phagocytic. That means that they're going to be able to engulf pathogens that are on or in the epidermis. Now coming on down to the thick portion of the skin, or the main portion of the skin, that's referred to as the dermis. So it lies beneath the epidermis. It's composed of dense connective tissue, and it's going to be made up of dense irregular connective tissue, and towards the top it's made up of some areolar connective tissue. This will contain lots of collagen fibers. These collagen fibers are going to give the skin strength and flexibility. And so you might notice as you start to age, these collagen fibers are going to start to break down and you won't have as many. So that's what causes skin to start sagging and not have as much resilience to it. Now going in with the chemical role, it is going to use things such as perspiration or sweating. It contains salt. That's going to create a hypertonic environment that's going to be inhospitable to microorganisms. Also, it's going to be slightly acidic. It's going to have a pH of around 5 to 5.5, which will inhibit bacterial growth. It's going to result from epithelial cells keratinization as they move towards the skin's surface, and it's going to cause that pH to drop. Also, it results from sebaceous glands secreting oils and waxes. These oils they secrete is going to be referred to as sebum. So that's oil on your skin. That's the proper name, sebum. Lysozymes are also going to be another thing here. These are going to be organelles inside of cells that contain enzymes that are able to destroy bacterial cells. Lots of times they might be acidic or they might just have the ability to go in and to break these different pathogens down. Another first line of defense is going to be your mucous membrane. So they're part of that innate immunity. So the role of the mucous membrane is that it's going to line the lumens of the respiratory, urinary, gastrointestinal, and reproductive tracts. Now, lumens are going to be openings in tubes, or openings or holes through tubes. They're going to play two roles. They have a mechanical role, and they're also going to go in and they're going to have a chemical role again. So within the mechanical role, it's going to be made up of two layers. The epithelium is going to be a thin outer covering of mucous membranes that's going to contain living cells that are going to be tightly packed together. Because of this, it's going to prevent entry of those pathogens. It's going to continually shed and carry away these microorganisms as it's shedding. So it can be shed within and you cough it out. If it's in the lungs or the trachea or the esophagus, it could be shed and go down to the stomach where it's going to be destroyed by stomach acid. So you're constantly shedding these and destroying them like your epidermis on your skin. It also is going to be made up of a deep connective layer that's going to lie beneath and support that epithelium. It's going to contain mucus, which is going to be a thick, sticky material that's going to be secreted by a specialized cell called a goblet cell. Now, there's going to be several functions to this. It's responsible for trapping dust and microorganisms, and that's a great one that we find within the respiratory tract, so you'll see that within the lungs, the trachea, the bronchi, and even the nasal passages. It helps to moisten and lubricate the mouth and esophagus, so that helps with the digestion. It's going to provide protective layers to prevent infection and damage to the epithelial cells. We're going to find that within the stomach. It's going to even have cilia on some of these cells, especially like in the respiratory tract. These are going to be short hair-like projections that are going to be found on cell membranes. And so they're responsible for like sweeping the potential pathogens that get trapped in the mucus. They're going to sweep them and cause them to either come up, so you cough them up and spit them out, hopefully, or sweep them up so then they go down the esophagus into the stomach and get destroyed by the stomach acid. And so you can see the cilia in this picture here that's going to be found between the different cells that are in this respiratory tract. Now, going in with the chemical role, it's going to have things such as lysozymes, antibodies, and acidity that's going to help. So with the lysozymes, that's going to be the lysosomes that are going to contain enzymes that destroy bacterial cell walls and help to break them down. It has antibodies. Those are going to immobilize these pathogens, make it so that they can't go in and infect any other cells. And acidity. That specific regions are going to be lined with mucous membranes are going to have very low pHs. For example, the stomach has a pH of about 1 to 2, and the vagina is going to have a pH of 4 to about 4.5. So both of these are going to be able to inhibit bacterial growth. Our next innate immunity defense is going to be a lacrimal apparatus. Now, your lacrimal duct is going to be found here that you see in the eye. And lots of times it's referred to as the nasolacrimal duct. So with the lacrimal glands, they're found in the upper portion of your eyelid, as you see in that first picture on the left. And once it releases the tears and the enzymes that are going to be in it, it's going to flow across the eye. It's going to empty into the lacrimal canal, which is going to be in the center portion of your eye. From there, it drains down, and it drains down to what's called the nasolacrimal duct. So that's why when you cry, lots of times your nose starts to run, because it's going to drain down through your nose. So with this, it produces and drains tears. It's going to blink, and when you blink, it's going to help to spread these tears and to wash the surface of the eye. And in those tears is going to contain lysozymes. So those are your enzymes that are destroying bacterial cells. So, role of normal microbiota. Microbial antagonisms are going to be normal microbiota that compete with potential pathogens. So these are going to be things that are normally found within your body or on your body. And normally they don't cause any type of infection unless they have no competition, and then they may cause problems. So 5 ways that they provide protection. Number 1, they help to consume nutrients. So the organisms that are bad for you have less nutrients to consume because they're competing with your normal microbiota. They also create an environment that's going to be unfavorable to other microorganisms. They stimulate the body's second line of defense. They're also going to promote overall health by providing vitamins to the host. For example, E. coli, which is found in the large intestine, is going to provide vitamin K for the body. And another thing, they're going to be able to compete to different binding sites that are going to be found on cell structures. So these pathogens must bind with cellular surfaces in order to get in and infect that cell and to cause damage or cause a disease. So some other first-line defenses, you have antimicrobial peptides. These are also known as defensins. They are present in the skin, the mucous membranes, and some white blood cells. So they're going to work by disrupting the cell membranes, and they're going to help to interrupt enzymic actions. So enzymatic actions are going to be interrupted so they can't perform their normal job. So some other chemicals and things and processes. Many organs are going to secrete chemicals with antimicrobial properties, such as stomach acid. It's going to prevent the growth of potential pathogens, and saliva. It's going to help to wash away microbes from the teeth, and it will also contain lysozymes as well. If you look at this chart below, you can see the different organs or the different areas within the body, and then you can see their function and how they're able to fight these different microbes. Now coming on down to the second line of defense. So those were all part of your first line of defense. Now we're going into the body's second line of defense. So with the second line of defense, it's going to operate when pathogens penetrate the skin or mucous membranes. It's going to be composed of cells and antimicrobial chemicals. Many are going to contain or originate in the blood. So first we're going to go in defense components of the blood. You're going to have plasma. Plasma is going to consist of water-containing electrolytes, dissolved gases, nutrients, and protective proteins such as clotting factors, complement proteins, and antibodies. Also, it's going to contain compounds that transport and store iron. This is going to play a role in defense by sequestering iron, making it unavailable to microorganisms. Defensive blood cells are going to be things such as leukocytes. They're going to be involved in defending against invaders. So there's 5 types of white blood cells that are going to be divided into 2 categories. The first, you're going to have what's called the granulocytes. This is the name that's given to them because their cytoplasm has a granular appearance. It looks like it has little grains throughout it. The first one that you're going to see is going to be a neutrophil. Now with the neutrophils, they are going to be, actually it's the one that's going to be in the center there, that bigger one. With the neutrophils, they're going to be the ones that make up about 60 to 70% of your white blood cells. They're the first to arrive at an infection site. What they're going to do is phagocytize bacteria, fungi, and some viruses. They're going to be highly mobile. So when they phagocytize a cell, that means that they're able to take and wrap themselves around that microbe, and they're able to bring that microbe into themselves and then destroy it using its lysozymes. Now the next one that we're going to talk about is going to be the eosinophil. So that's going to be to the right of the neutrophil. This is going to make up about 2 to 4% of your white blood cells, and it's going to help to combat moderate allergic reactions, and it helps to defend against parasitic worm infections. Next one is going to be the one to the left of the neutrophil. It's going to be your basophil. It makes up about 0.5 to 1% of white blood cells. It's going to release histamine and heparin, which are going to promote inflammation. So these are the ones that will help with inflammation and help to get more blood cells to an area. Next, we're going to go to what's known as the agranulocytes. This means the cytoplasm does not have any granular appearance to it. And so these are going to be your monocytes, which you're going to see over on the right-hand side. This makes up about 3 to 8% of your white blood cells. They become macrophages that phagocytize bacteria, dead cells, and other debris. They're also going to be highly mobile and able to move around. Next, we're going to go to the lymphocytes. That's going to be beside the monocyte all the way at the end on the right-hand side. These make up about 20 to 25% of white blood cells. They're going to be important in adaptive immunity. So these will be your T cells and your B cells. T cells will directly attach to microorganisms and tumor cells and transplanted cells. B cells are going to be important because they're going to produce antibodies that are going to go out and they're going to attack these foreign molecules that aren't recognized or don't belong in your body. Now, if you're sick and you go to the doctor, they may order a differential white blood cell count. What this is going to do is it indicates the percentage of various types of leukocytes that are going to be present in the blood. It can signal signs of different diseases or infections. For example, an increased eosinophil count is going to be related to things like allergies or parasitic worm infections. If you have increased leukocytes and neutrophils, that means that you could have a bacterial infection or disease. And if you have increased lymphocytes, it could be a viral infection. Now, we've talked about phagocytosis a little bit, but here's a good representation, a good diagram here. So phagocytes are going to be cells that are capable of phagocytosis. So they divide into 6 stages. First, you have chemotaxis, which is going to be positive. It's going to be a movement of cells either towards a chemical stimulus, and it's going to have what's called a chemotactic factor. So these factors are going to be chemicals that attract phagocytic white blood cells to the site of damage or the site of a bacterial, virus, whatever microbial invasion there may be. Second, you have adherence. This is where these phagocytes are going to attach to the pathogens. Third is ingestion. They're going to use pseudopodia. That's where they're going to have extensions of their cell membrane that are going to surround, ingest, and bring in this microbe. So it forms what's called a phagosome that you see here. This phagosome basically looks like a lysosome, or looks like a little vacuole. It's surrounded by the cell membrane, and it's bringing it in to the body of that cell, kind of like a food vesicle. And so it brings it into that white blood cell. Fourth, the phagosome's maturation and microbial killing is going to involve lysosomes that are injecting digestive enzymes to form acidic environments directly into these phagosomes here. So that's going to help to kill them. And then the final step will be elimination. That's where the microorganisms are going to remain in there until they're eliminated and then gotten rid of through exocytosis. So it kind of spits out the eaten up or broken down parts. Non-phagocytic killing could be things like eosinophils, where they go in and they attack parasitic helminths. They're going to do this by getting close to these worms, and they're going to secrete toxins that are going to weaken or kill the worm that's going to be in there. They attack bacteria by coating them with a mitochondrial DNA, and that's going to be investigated, and it's going to go into actually breaking down that bacteria. But they're still investigating on how it works because they're not quite sure to be completely sure of how it's happening, basically. Next, we have the natural killer lymphocytes. I like these. These are the NKs, is how they're usually represented or talked about. These are going to secrete toxins onto the surface of virally infected cells and help to destroy them. So basically, they cause holes in the cell membrane of those cells. And they'll go around and they look for cancerous cells within your body, and they actually put holes in those cancerous cells and destroy them. So you have natural killers, or NKs, constantly circulating through your blood and through your body tissues. And once they come across a cell that they pick up that something's not quite right and it may be cancerous, they'll go ahead and they'll start to digest the cell membrane and destroy that cell. Next is going to be the neutrophils. They're going to produce chemicals that are going to kill nearby invaders, and they also produce neutrophil extracellular traps. These are going to be extracellular fibers that are going to be able to bind and kill bacteria. So nonspecific chemical defenses. We're going to have chemicals that assist phagocytic cells in 2 ways. Directly attacking the pathogens, or by enhancing other features of innate immunity. 4 different types of chemicals are going to help. We have what's called toll-like receptors. These are integral membrane proteins that are produced by phagocytic cells. They're going to go in and bind to pathogen-associated microbial patterns, which are called PAMPs. The microbial structures here are going to be shared by bacterial and viral pathogens, but they're going to be absent in humans, including the peptidoglycan. It initiates 3 different defense responses. The first one, apoptosis. That's where a cell commits cell suicide or cell death. Second, secretion of inflammatory chemicals. And then thirdly, it's going to produce adaptive immunity responses that are going to help to stimulate adaptive immunity to come in. Next, we have NOD proteins and interferons. So with the NOD proteins, these have receptors for the PAMPs that are going to be found in the interior of the cell. It's going to trigger a set of defensive responses that are going to be similar to those TRLs that we just saw. With the interferons, these are going to be released by a host cell to non-specifically inhibit the spread of this viral infection. If it's viral infection, it's going to be produced by several different types of cells, including things like lymphocytes, monocytes, and macrophages. Now, there's going to be two different types of interferons. Type 1 is going to be called alpha and beta interferons, and they're going to provide antiviral action and also act against tumor cells. Type 2 are going to be the gamma cells or the gamma types, and they attract phagocytes to areas of infection and increase their activity. Next, we're going to have our complement proteins. This is going to be a set of proteins that's going to be produced by macrophages that act as chemotactic attractants. So they are indirectly triggers of inflammation and of fever. They're going to be activated in three ways. You have your classical pathway, that's where it's activated by antibodies. You're going to have the alternate pathway, that's where it's activated by pathogens or pathogenic products. And you have what's called a lectin pathway. It's going to be activated by microbial polysaccharides that are going to bind to activation molecules. So inactive or of complements, these are going to be cellular proteins that are going to bind and break down to activated complement proteins, and they allow the body to withstand this complement. So the complement activation will result in foreign cell lysis. Now, lysis is going to be where it can go in and cause that cell to break up and become destroyed. Basically, it breaks it into pieces. Now, going into inflammation. It's going to be defined as a general nonspecific response to tissue damage resulting from a variety of causes, including infection with pathogens. It's going to be characterized by things like redness, heat, swelling, and even pain. The first type that we're going to talk about out of two types is going to be acute inflammation. That's inflammation that's going to develop quickly and short-lived and beneficial in destroying pathogens. It occurs in three different stages. The first stage is going to be dilation and increased blood vessel permeability. Here, the body's initial response to injury or invasion has started with this pathogen coming in. Blood clotting is going to be released by bradykinin. That's a protein mediator of inflammation. Next, you're going to have these macrophages release inflammatory chemicals, things like prostaglandins and leukotrienes. They're going to be responsible for increasing permeability of small veins. You also have a histamine that's going to be released by cells called basophils. Know that basophils release histamine. They're mast cells, basically. And they're also going to be released by things like platelets. And all of this is going to happen in connective tissue cells there. Second, you're going to have migration of phagocytes. So here, the monocytes and neutrophils are going to arrive on site of the infection. Margination, is going to cause them to stick to the blood vessel walls, and then it's going to squeeze it into the site of the infection and destroy the pathogen through phagocytosis. So you see it sticking to the sides of the cell walls and then squeezing through those little openings. Third is going to be your tissue repair. That's going to involve the delivery of extra nutrients and oxygen to the site. Certain tissues like skin and mucous membranes are going to be repaired very quickly, while other tissues are going to require things such as fibroblasts. And that's going to create scar tissue when the fibroblasts come in. Cardiac muscle and brain tissue cannot be repaired, so these will be filled in with this fibroblast and create scar tissue. So here's another view that's showing you the migration of those phagocytes as they're kind of getting through those cells that are making up the blood vessels there. And so you see how it can squeeze into the tissue, the little elongated small particles, that's going to be your bacterium. And so it shows how they can squeeze through and go after the bacterium. And this shows your tissue defense. So we see how the bacteria is getting in through that cutter, that opening. You see how those damaged cells are going to release those prostaglandins and leukotrienes and histamine. Next, you're going to see how those prostaglandins and leukotrienes make the vessel then more permeable. From there, the macrophages and neutrophils are able to squeeze through, coming on down to the left corner. It's going to increase the permeability. It's going to allow all of these different antimicrobial chemicals and clotting proteins to come in. And eventually, you're going to have a blood clot forming. You're going to have more phagocytic migration. of the site of this bacteria here. Eventually you have the accumulation of that damaged tissue and leukocytes and pus there, and then you're going to have those undifferentiated stem cells start to fill in that area. Now, with this, you could have chronic inflammation. That's going to be inflammation that develops slowly but lasts a long time and can cause damage or death to tissues, or it could result in a disease. Now, with the fever, this is going to be defined as a body temperature that's going to be above 37 degrees Celsius or 98.6 degrees Fahrenheit. It's going to occur as a result of chemicals called pyrogens, so you need to know that. Pyrogens are chemicals that cause fevers. It's going to be triggered by the hypothalamus, or what we call the hypothalamic thermostat, and it has to reset at a higher temperature because it's going to identify that there's something that needs to be taken care of, and usually that higher temperature can alleviate it. So 5 functions. It's going to enhance the effects of interference. Second, it's going to inhibit the growth of some microorganisms. Third, it's going to enhance the performance of phagocytes. Fourth, it's going to enhance the activity of cells of specific immunity. And then fifth, it's going to enhance the process of tissue repair. There are going to be some unpleasant side effects, though, of a fever. It could cause you malice. You could be tired. You could be grumpy. You're not feeling very well. You have body aches, and you might be very weak. The fever, if it rises too high, it can start to denature proteins within the body, and it can even inhibit some nerve impulses, so it can result in seizures and even death. Now we're moving from innate immunity over to adaptive immunity. Adaptive immunity is going to be the body's ability to recognize and defend itself against distinct invaders and their products. Before, the body was just defending itself against whatever came in. This is actually going to adapt according to what it's being invaded by. It's going to have 5 attributes. First is specificity. That's going to be the response that is going to be precisely tailored reactions against specific attackers. Next, we have inducibility. That's going to be where cells act only in the response to specific pathogens. Third, we have clonality. That's where cells are going to proliferate and they're going to be able to form generations of nearly identical cells called clones. Fourth, we have unresponsiveness to self. That's where it has to be able to go in and not act against normal body cells. So it's got to learn to be immunocompetent. Only about 2 cells actually make it past this test and become immunocompetent and are able to go out and destroy things that do not belong to the body. And then fifth is memory. That's a response that's going to be a faster and more effective response in the future when it encounters the same particular pathogen. Now coming on down, we have the lymphatic system and of tissues and organs. With the lymphatic system, vessels, and the flow of lymph, it's going to be able to conduct that flow. You're going to see that all these vessels that are here in green are going to be our lymphatic vessels. Now within these lymphatic vessels, it's going to contain a colorless, watery liquid similar to blood plasma that's going to be referred to as lymph. It's going to arise from leaked blood vessels that surround the tissues. So what happens is this is basically plasma. It's going to leak out, go into the surrounding interstitial tissues. It's going to carry things like nutrients and oxygen and bathe those cells in these products, and it's going to absorb what it needs, and then that fluid needs to be able to get back into the flow or the circulatory system. So it's going to empty out into these lymphatic vessels. And so that's what you see happening in that top picture there, where you see the tissue and you see the arrows coming into those green vessels. So it's leaving that tissue, going into these vessels, and then will be carried back up to the vessels that enter the heart so it can re-enter the circulatory fluid or re-enter the plasma basically. And so you're going to have 2 different types of lymphoid organs. They're going to be directly involved in specific immunity and have 2 different types. Here we have what's called the primary lymphoid organs. Those are going to be things such as red bone marrow and thymus. And then you're going to have your secondary lymphoid organs. Those are going to be your lymph nodes, your spleen, your tonsils, and your MALT, which is going to be your mucosa-associated lymphoid tissues. So antigens are going to be very important. These are going to be portions of cells, viruses, molecules that the body recognizes as foreign. So antigen properties. First, they have what's called epitopes. That's going to be a 3D shape of the antigen region that the immune system recognizes. Second is the effective antigen. It's going to be large, complex, stable, degradable, and foreign. This includes things like proteins, carbohydrates, lipids, and bacterial DNA. Now, there's going to be 3 types of antigens that we'll see. You have what's called an exogenous antigen. That's going to secrete or it has components of microbes. This will include things like toxins, cell walls, cell membranes, flagella, and pili. Second type is going to be called an endogenous antigen. These are antigens that are produced when protozoa, fungi, bacteria, and viruses reproduce inside body cells. These can only be responded to if the antigens are going to be incorporated into the cell membrane. And then the third type, which is called an autoantigen. Those are molecules that are going to be found on an individual's normal, uninfected cell. So it needs to learn self-tolerance. So the immune system has to recognize the antigens that are found on your normal body cells, so it does not attack these cells. It doesn't see them as foreign. So here we have B lymphocytes, or B cells, and antibodies. So we have specificity to B cell receptors. These are going to be found in places like the spleen, MALT, and primarily in lymph nodes. These are going to arise and mature in the red bone marrow. The major function here is to secrete antibodies. The specificity receptor cell is going to be an antibody-like protein, that's not cell, but protein, is going to be an antibody-like protein that's going to be found in the cell membrane. It's a complementary to that antigen epitope. Its antigenic epitope binds to the BCR, which is going to be the B cell stimulating itself when it binds here, and becomes a plasma cell. Once it's a plasma cell, it's going to secrete the soluble antibody molecules. So, with specificity and antibody structure, these immunoglobulins are going to be similar to BCRs. They're going to have identical antigen binding sites and antigen specifics as the BCR do. So there's going to be several different antibody functions. We're going to have 6 main functions here. We have activation of complement and inflammation. You're going to have neutralization of toxins. Third, you have what's called opsonization. This is where antibodies are going to act as molecules that stimulate phagocytosis. These are going to be opsonins. Fourth, you have the killing by oxidation. That's where antibodies with catalytic properties will kill bacteria directly. Fifth, you have what's called agglutination. That's where you're going to have the clumping of antibody molecules with microbial cells. This is going to hinder the activity of those pathogens and increase phagocytosis. And then lastly, sixth, you're going to have antibody-dependent cellular cytotoxicity, which is ADCC. That's going to stimulate a phagocytosis by antibodies causing apoptosis or cellular death. Now you're going to have different classes of antibodies, and you can enlarge this on your regular PowerPoint, but we're going to discuss these 5 classes. First, you have what's called immunoglobulin G, or IgG. These are going to be predominant antibodies that are going to be found in the bloodstream. They're primary defenders against invading bacteria. Second, you have immunoglobulin M, or IgM. Oh, I didn't mean to turn that with my finger, sorry. These are going to be the predominant antibodies that are produced during first immune responses. Third, you're going to have immunoglobulin A, or IgA. They are associated with various body secretions. Fourth, you have immunoglobulin E, or IgE. They're involved in responses to things like parasitic infections and allergies. And fifthly, you have immunoglobulin D, or IgD. This is going to be found in some animals as B cell receptors, and right now we're not quite sure what their function is. Their function's unknown. Now going into T lymphocytes, or what we call T cells. The T lymphocytes are going to be produced in the red bone marrow, and they mature in the thymus, so that's where they get the T from. They're going to circulate in the lymph and the blood and migrate to the lymph nodes and spleen. T cell receptors are going to be found on the cell membrane, and they're going to bind to sites that are going to be complementary to the epitopes. Specific T cell receptors, or types of, well, let's go back to the specific ones first. They're not going to be recognized as epitopes directly. They only bind to these epitopes associated with the MHC protein. And so that's going to help hold the position of the epitopes for presentation to the T cells. So this is going to help to act primarily against cells that are going to harbor intracellular pathogens. Now with the different types of T lymphocytes, you're going to have three types that we're going to talk about. We have the cytotoxic T cell, which is going to directly kill infected cells and they're going to kill cancer cells. We have the helper T cells. With the helper T cells, these are going to regulate the activity of B cells and cytotoxic T cells. And then we have what's called regulatory T cells. These are going to repress adaptive immune response and prevent autoimmune diseases. So clonal deletion is going to be a process by which lymphocytes respond to autoantigens and are selectively killed by apoptosis. So basically the body eliminates self-reactive lymphocytes. The clonal deletion of T cells is going to occur in the thymus. So we'll have thymus cells that are going to process and present all body autoantigens to young T cells. Next, we have the clonal deletion of B cells. This is going to occur in the bone marrow. So think of B cells, bone marrow, T cells, thymus. So this one occurs in the bone marrow. And what happens is the red bone marrow process, and they present all the body's autoantigens to these young B cells, and they test them to see if they're going to be reactive or not. So if the T cells are reactive or if the B cells are reactive, then they have to be deleted or destroyed through apoptosis. Autoimmune disease is going to be when the self cannot tolerate these T cells, B cells. They don't tolerate the cell's self-antigens. And so what happens is that self-tolerance is impaired and it goes in, it begins to make autoantibodies or cytotoxic T cells against normal body cells and then starts to damage and destroy normal healthy tissue. So with immune responses, we have the cytokines. These are going to be soluble regulatory proteins that act as intracellular signals. They're going to be released by certain body cells like the kidneys, skin cells, and immune cells. You're going to have 5 types of cytokines of the immune system. So looking here, it shows you a list of these. You have your interleukins. These are going to signal among leukocytes. You have your interferons. They're going to be antiviral proteins that are also going to act as cytokines. You have growth factors. These are going to stimulate stem cells to start dividing and go through mitosis. You have tumor necrosis factor. These are going to kill tumor cells and regulate the immune response to them. And then fifth, you have the chemokines. These are going to be a signal to the leukocytes to move to the site of inflammation or infection and activate the leukocytes. So some roles of the major histocompatibility complex. We talked about MHC a little bit earlier. That's what it stands for, major histocompatibility complex. It's going to be a cluster of genes located on each copy of a chromosome, and it's going to code for major histocompatibility antigens. This is going to be present in cytoplasmic membranes of various cells and is going to hold and position these antigenic determinants for presentation to the T cells. So they hold them in place so the T cells can come and take a look at them. Antigens will then bind to this antigen and it's going to bind into that groove that you see there. So that antigen binding groove is what it's looking for. And you're going to have 2 classes of these proteins. You'll have what's called the class 1 MHC molecule and the class 2 MHC molecule. So the class 1 is going to be found on the cell membrane of all nucleated cells. So that means all cells that have a nucleus. The class 2 is going to be found only on B cells and on antigen-presenting cells. So these antigen-presenting cells basically are going to be macrophages or dendritic cells that you're going to find this MHC2 on. Now with antigen processing, that must be processed before the MHC proteins can display these epitopes. There's 2 types of antigen processing. You have processing of endogenous antigens. These are going to be the polypeptides that are going to be catabolized internally, resulting in a peptide epitope, which is going to bind to the MHC class 1 molecule. From there, it's going to be exported to the cell membrane of the nucleated cell. The next type you see here is going to be the exogenous antigens processing. So here the antigen processing cell is going to be enzymatically catabolized, and so it's going to go in and it's going to break it down using enzymes and molecules, and it's going to produce peptide epitopes, which are then going to bind to the MHC class II molecules. These are going to be exported to the cell membrane of the antigen presenting cell. Now we're going into what we call the cell-mediated immune response. So this is going to be a response to intracellular pathogens and abnormal body cells. It's the most common intracellular pathogen that it's going to come across is going to be a virus. It's effective against cancer cells though, also intracellular protozoa, and intracellular bacteria. But viruses are what it works the best with. So activation of T cell clones and their functions, we're going to have the adaptive immune response is going to be initiated from the lymphoid organs, and it's going to do this in 4 steps. You're going to have antigen presentation, which we just saw how that happened, and it's going to occur in nearby lymph nodes. The helper T cell differentiation is going to help to assist in creating links between antigen presenting cells and T cells. You have clonal expansion. That's going to be a process where it's going to activate lymphocytes to divide from memory T cells and make more of these memory T cells. So basically clone itself and make many of itself. And then you have self-stimulation. That's where daughter cells are going to be activated and produce receptors and signals that are going to allow them to leave the lymph node and to attack virally infected cells. Memory T cells are going to be a type of T cell that's going to be present in the lymphoid tissue for months or even years. Immediately, they function upon subsequent contact with the epitope of specific to its TCR. Memory T cell response is going to be more rapid and more effective than primary responses. With T cell regulation, it's going to be a regulation needed to prevent T cell response to autoantigens, so you don't want to respond to yourself. T cells are going to require additional signals from an antigen-presenting cell. So if it has no signal, basically there's going to be no response. But if there is a signal, then that T cell will go in and it will respond. Now, humoral immune responses, we're going to have induced response against exogenous pathogens. It's going to be activated only in response to specific pathogens. And there's going to be 2 different types of humoral response. The T-independent humoral response immunity is going to involve the T-independent antigens. These are going to be antigens that are going to be large and contain repeating epitopes. These antigens can be processed by the B cells only, and they do not involve T cells. They repeat subunits of antigens that allow cross-links between numerous BCRs. They induce fast responses, but response is going to be weak, and it disappears fairly quickly and induces little immunological memory. Now, the next type is going to be the T-dependent humoral immunity. This is going to involve T-dependent antigens. They are going to be smaller antigens that are going to lack repetitive epitopes. These antigens can be processed by B cells. It does not involve the T cell or helper T cell, basically, with this one. And it's going to have 4 interactions between antigen-presenting cells and the helper T cells that help to mediate these cytokines. The first is antigen presentation for the TH activation and cloning. Second, you're going to have differentiation of that helper T cell into the TH2 cells. Third, you're going to have clonal selection. Sorry, going back here. Didn't mean to go forward. You're going to have your clonal selection. That's where the B cell with the BCRs are going to be complementary to the epitope, and that will be recognized. And fourth, you have activation of B cells by the TH2 cell. That's going to result in rapid B cell proliferation, or the making of lots of those. Now, coming into the memory B cells, these are going to be cells with BCRs that are going to be complementary to specific antigen triggering production. They persist in the lymphoid tissues, that's where you're going to find them, and they initiate antibody production if antigens are going to be encountered again. The primary immune response is going to be slow and limited in response to the first encounter with an unfamiliar antigen. But then the secondary immune response is going to be enhanced immune responses following a second contact with that antigen. And lastly, you have that memory response, which is going to be a rapid and enhanced immune response to subsequent encounters with familiar antigens. So when it comes to immunity, you're going to have several different types of acquired immunity. You have naturally acquired active immunity. That's where the body responds to exposure to antigens by mounting specific immune responses. Naturally acquired passive immunity is where things like a fetus, a newborn, or a child will receive antibodies across the placenta or within the breast milk that it receives from its mother. Third, you have artificial acquired immunity, and that's going to be an active immunity. The body's going to receive antigens by injection, as with things like vaccinations, and it's going to mount a specific immune response. And then lastly, you're going to have artificially acquired passive immunotherapy. This is where the body's going to receive something via like an injection, where it's going to have preformed antibodies in antitoxins or antisera. These are going to be different antiserums, kind of like they were making for COVID at a time. These can go in and they can destroy these fast-acting and potentially fatal pathogens. And so that's one that is going to be created or gotten from somebody else and is injected directly into you. Now, a brief history of immunization. In the 12th century in China, infected young children with particles of ground smallpox scabs were going to be the first things that were used. In this, it was a procedure that they called virilation. It spread to England and America, but stopped due to the risk of death because putting these particles into the human body could cause a full-blown infection and could result in death. But lots of times it was just enough to give them some immunity. Now in 1796, Edward Jenner, that you see here down in the lower left-hand corner, discovered the process of vaccination. He learned that the protection against smallpox conferred by inoculation with cowpox was very, very beneficial. Later on, in 1879, we have Louis Pasteur. He's going to make the first effective vaccination that they were able to start using. By the end of the 20th century, vaccines have been developed for several potentially fatal infectious diseases. The reduction in infections of these diseases and mortality worldwide actually helped people be able to survive these things that normally they couldn't survive before. Vaccines today are developing. Now we have recently developed the most recent one, which was the COVID vaccine. Developing nations, unfortunately, though, usually don't receive these vaccinations. Effective vaccines are going to be not developed completely now, even for some pathogens that we have. Like they've still been trying to work on one for HIV for quite some time, well over 20 or 30 years. So there's some that they cannot vaccinate against, and then there's some that are in the process that they're working on. Now, different types of vaccination. Now, immunization is going to be developed from the administered antigens, so patients actively mount a protective immune response. So with these, we'll have 5 different types of vaccines. You have what's called attenuated or live vaccines. These are going to use the pathogens with reduced virulence. They may result in you actually getting that illness, but usually it's going to be a very mild case. They stimulate strong immune responses and could retain enough residual virulence to cause that disease. That is kind of a disadvantage, like I said, but usually it's going to be very low. Second type is going to be an inactive vaccine. That involves killed microbes. 2 different types of microbes that are going to be used here are going to be whole agent vaccines. They are deactivated whole microbes, or subunit vaccines where they just have microbial fragments. These don't provide antigenic molecules to stimulate the immune response, and boosters are usually used to achieve immunity. Adjuvants, these are going to be chemicals that are also added to increase effective antigenicity to it. Third, we're going to have what's called toxoid vaccines. These are chemically or thermally modified toxins. They are multiple dose required vaccines that you take usually during childhood with reinoculation about every 10 years. Fourth, you have a combination, which is going to be administration of antigens from several different pathogens. An example of this one would be the MMR, measles, mumps, and rubella vaccine. And then the 5th, we have what's called recombinant vaccines. These are going to be made using recombinant techniques or altering the genes. These make vaccines more effective, it makes them cheaper, and it makes them safer. And vaccine manufacturers are going to mass produce these vaccines Basically, they're going to grow microbes in culture vessels and grow a lot of them all at once. Also, they'll have viruses that are cultured inside of chicken eggs that they even use. Now, there is a recommended immunization chart or list that you can view right here, and it shows you the age and the different vaccines that are recommended. Vaccine safety. Well, common problems with vaccines is that sometimes there is mild toxicity. You could even have anaphylactic shock. It could be a residual virulence from live viruses that you may acquire, or you could have allergies to possibly the vaccine or the substances within the vaccine, like the things that keep it shelf stable. Passive immunotherapy is going to involve administration of preformed antibodies to a patient. These preformed antibodies are going to be obtained from people or animals having experienced a natural case of the disease where it actively was immunized against, or it was actively immunized against it, This is going to provide rapid protection against a recent infection, but it doesn't require the body to mount its own response. The antibodies are going to be immediately available to bind to these antigens. Unfortunately, there are limitations. You could have allergic reactions. The antibodies can degrade relatively quickly so they can break down. The body doesn't produce more memory B cells, so the patient's not going to be protected from subsequent infections after this. And so that could all be a problem. So that's going to finish us up for Unit 10. I'll see you back here for Unit 11.